Okay, so we are going to continue on with cytoskeleton here.
So I was just thinking about you right here.
That kind of works. And we just got done talking about microtubules, and now we're going to move on to microfilaments. So just like the microtubules, microfilaments are going to be made up of a polymerization of multiple proteins, but instead of having a heterodimer, we just have a single polypeptide that is making up the individual proteins, and in this case, that is actin. Before I forget things. So, actin is actually a fairly large protein. It's made up of 375 amino acids, which doesn't make it incredibly large. And we have two different forms of actin. First off, we have G-actin, which is just an individual peptide of actin. But we also have this thing called F-actin, that is the polymerized form of G-actin. So G-actin, the individual thing, and F-actin is the polymerized form. So to make a whole microfilament, we have two F-actin strands then that form a double helix, and that is then the full microfilament.
Is it recording?
Yes, I am recording. I decided not to do that anymore. This chair. So that looks something like this. We have the individual, these were the individual peptides of actin, and then we have, or polypeptides of actin, and then we have one F-actin here, we have the second F-actin there, and then they're all nice and interwoven, forming double helix, and that is a microfilament. We generally have these, I'm going to sit down, that's nicer than standing up. We generally have one end of these bound to the membrane, and again, just like what we had before, we have a plus and a minus end. And that's actually determined based on the structure. You're messing with my hat. You should drop your orange if you're messing with my hat. Here we go. It's a very nice hat. Oh, you don't mess with nice hats. Okay. Anyway, so one end of the microfilament, the minus end, which is determined based on the structure of the G-actin that makes up the individual monomers in there. I'm not going to have you guys, you don't need to know exactly how you determine that. What you do need to know is that the minus end is attached, generally bound to the membrane. And then assembly and disassembly occurs at the plus end. So this is kind of similar to what we saw in microtubules, is that assembly, disassembly, or polymerization preferentially happens at the plus end. Remember, in microtubules, it's not only happening at the plus end, it's just happening about twice the rate at the plus end. In this case, it begins with the minus end bound to the membrane, and then the plus end is where our assembly and disassembly is going to happen. And we can control polymerization by several different factors. And I guess the first one is kind of obvious. The concentration of G-actin that we have actually in the cell or in the local area is going to control polymerization. If you don't have G-actin to make your microfilament, you're not going to make a microfilament. And if you don't have very much, you can't make it very fast. So that's one of the basic controls that we have right there. Also, this takes energy. We can't just do this without energy, so the ATP concentration, or the availability of ATP, also controls the polymerization of microfilaments. We need 1 ATP per G-actin monomer that we add to the entire string. Okay, that got boring. And then additionally, we also need the enzymes that actually do this polymerization have cofactors that are important as well. One principal one in this is magnesium. So the presence of magnesium ions is also important to control the polymerization of actin as well. So, or polymerization of microfilaments. So if you don't have that, you cannot polymerize microfilaments. Yes, here.
Is there a reason G-actin is more important than the other F-actin?
Well, the F-actin is just the polymerized G-actin. So it kind of leads to, if you don't have G-actin, you can't make the F-actin. So yes, by consequence, if you don't have F-actin, you also can't make these. But really, it's not. Polymerization doesn't happen by, we get a whole F-actin strand and a whole another F-actin strand and twirl them about. It's added by, we have the, actually let me go back. Might be easiest to illustrate this by going back here. Instead of me drawing this, since I already took the time to draw it. If this was our end, so we're bound to a membrane over here, imagine, and we have this, we're not going to like unwind this, chunk on another chunk of F-actin. We're going to add individual monomers to the ends of each of these F-actin, and so it really is the G-actin itself. I mean, given you need to make it into F-actin, but you're not making it into F-actin first and then taking two pieces of F-actin and polymerizing them. Does that make sense?
So F-actin doesn't exist as just by itself?
It does, but in cases that we're not going to really talk about here. And in microfilament polymerization, it doesn't go through a stage of just being F-actin. Does that make sense? Okay. Is that overly confusing?
No.
Okay. Because sometimes I think I say things in ways that are more confusing than they really need to be. And I'm sorry for that. It makes me sad inside. But not that sad inside. I work on it. Okay. Just like we had maps before, I often rant on biologists making things more confusing than they need to be. And this, they actually did a little bit better job. They could have called them also maps, microfilament associated proteins, but in this case we call them MBPs, microfilament binding proteins, and these are going to regulate the growth and degradation of the microfilaments. It doesn't really help that much. And many of these require calcium to function. And you need different concentrations for different kinds of MBPs. And so you can control which MBPs are predominantly working by concentrations of calcium. So we also need, although it wasn't strictly said before, we also need calcium for many of the functions that are going to be carried out by the microfilaments to actually act. And yeah, let's leave it at that for now. Also, we have capping proteins. And so at some point, maybe that we want to stop polymerizing at some point. Like, okay, this is long enough now, we need to stop there. However, if you just leave the microfilament by itself without anything there to stop polymerize or kind of stabilize the ends become very unstable and they are prone to disassembly. So we need something to stabilize that end, and that's what capping proteins are for. We have two of them, fragment and brevin, that are predominant, and these bind to the plus end, again the side that is being polymerized, and we're going to stop elongation by capping it there. I didn't give you this. Did I give you the stamp on? Here's this. See, this is how my mind thinks. It's like it bounces around to different things I'm supposed to be doing. I should do this with all my handing back. It's like lecture a little bit, hand back a couple things, lecture a little bit, hand back a couple things. Are you sure I didn't give you back your Scantron? Yeah. Oh, you did get it. Okay, my bad. So I'll let you a couple more slides, and I'll hand some more things back to someone, I'm sure, because Okay, so one of the principal things that we talked about in controlling the polymerization of microfilaments is going to be the availability of the concentration of G-actin. And so actually we have certain things that will sequester the G-actin and prevent polymerization of microfilaments. Kind of makes intuitive, straightforward sense. And these will bind to the GTP, or excuse me, not GTP, G-actin. And therefore, you're preventing it from undergoing polymerization, and therefore we're stopping it from actually continuing on and doing any sort of elongation. Okay, so let's talk about a few different functions of microfilaments. And I'm just, for this moment, I'm going to tell you about the non-muscular functions. There is you'll take a lot of time in another course if you want to learn about the muscular functions. Anyone want to take a guess what that course would be? Elephants, yes! Wow, you guys are good. So we end up talking a lot about how the muscular functions of microfilaments and how that actually produces a muscle contraction in animal phys, but for this moment we're going to just keep it to the non-muscular functions. And so, for instance, one thing here is another thing that I was talking about earlier, this sometimes happens with microtubules, is that we actually use microfilaments to locate or kind of bind to enzymes and hold them into a certain position in the cell. And to hold certain enzyme complexes together, but then also enzymatic pathways, you want to have each of the additional enzymes in close proximity to each other, so we can hold those in close proximity by binding them to a microfilament. Also, microvilli structure. This is something like, for instance, in your intestines and other things, where you have small projections of a cell, they need to hold fairly rigid structure. You need that to happen, so we can use microfilaments to actually provide that structure. And so what we have here is vilin, which is at low calcium concentrations. We get cross-linking between actin. So different actin microfilaments will actually bind to each other. If we raise the calcium concentration, those binds will break. So actually this is one thing that happens during diarrhea, is during a dysregulation of your ends of your small and your large intestine. You actually get, because of other things that are being dysregulated and inability to regulate calcium stores build up in the marginal cells in the intestines. This breaks down the microvilli. This means that you have the number of microvilli or their structure being reduced, and this is bad. And one of the major things that you need microvilli for is because you generally have a lot of water in the things that you eat. The first thing that you notice is an inability to absorb water out of your digestive fluids as they pass through your intestines, and then you get things like diarrhea. This can also cause problems with other absorption as well, because you're not doing a really good job absorbing anything at that point. So yeah, add that in there, because Christmas time. Who doesn't want to hear about moving on? Yep. Also, these are a pathway for movement for molecular motors. We've talked a little bit about this with microtubules and the kinesin and the dynein. This also happens. The one that is very particularly important here is myosin. Myosin is a molecular motor that's very similar in function and operation to kinesin, and it is going to take a slightly larger step than what kinesin does. So we're looking at about 37 nanometers step for each of them. And so this is another thing that, again, looking at Muscle contraction is a primary molecular motor that's operating in muscle contraction. But this is not, muscle contraction is not the only time in which myosin is used on microfilaments. We also have cell migration and cell elongation. So essentially changing the shape of the cell, but then also using it to propel the cell somewhat. Microfilaments are using those functions as well. Save time, I'm not going to really talk too much about adhesion fibers and lamellipodium for the moment, so don't worry about those. Are there any questions on microfilaments before we move on? Yes?
You mentioned myosin, same as, or similar to kinesin rather?
Yeah. In general, broad strokes.
So then my question is, the dynein, what is the difference between that?
Okay, so it's also fairly similar to dynein as well, but I didn't go as much into detail on dynein, so I didn't compare it directly to that, because essentially all these molecular modes in very broad strokes act similar to the function that I showed you for kinesin, so I'm just comparing them back to that. Yeah. But all three of those work relatively similarly. Yeah. I mean, if you really got into it, there's actually fairly large differences, but it's not particularly important at this moment that what those are. But essentially we have the same thing, where we have a walking motion, we have ATP that drives the individual steps of that motion. Any other questions on the microfilaments? Yeah, Ben.
So microfilaments are smaller than microtubules, right?
Yes.
So would there be more of them in the cell?
I don't know, actually, if there's more of them in the cell in total. Because each of them has particular roles that they carry out, and we're not just jam-packing them in there to their capacity. It's not like you need... So I'm not exactly sure, to be totally honest with you. I don't know which you have more of in the cell. And it depends on what you mean more. If you were just to look at a total amount of them by weight, would be different than counting up the actual fibers.
I was talking about counting up the actual fibers.
How many fibers? And in that case, I mean, it doesn't actually change. I still don't know. But one of the things that would be hard about measuring that is both of these are really dynamic. And so you are creating and destroying these things often. And so if you come in and talk to me tomorrow, a good chunk of the microtubules and microfilaments that existed inside of you today will no longer actually be like cohesive things. Those monomers will still be recycled and used, but the individual ones are recycling very, very quickly. Which still, I mean, you can still probably count them up somehow. I don't know exactly what the counts would be relative. Cynthia, did you have a question? Oh, okay. Any other questions? That I can tell you, I don't know. Okay, let's talk about the third different one here. We are now going to talk about intermediate filaments, which, hey, they're actually somewhat intuitively named here because in size, particularly in diameter, they're somewhat intermediate between microfilaments and microtubules. It's kind of interesting because microtubules, despite having the word micro in them, are the largest of these. Intermediate is then the intermediate one, and then the microfilaments are the smallest. So these are about 10 nanometers in diameter, which again, yeah, kind of an intermediate size. Their structure, however, is going to be significantly different than the other two. Okay, so the previous ones that we talked about, we have these individual almost, I've been demonstrating them as spherical, but they're in real life they're somewhat globular monomers that then get polymerized to make some sort of filament. It's going to be somewhat different than what we're looking at now. So there's actually a lot of different individual proteins that end up making up intermediate filaments, and different combinations of these or different uses of the different proteins that make them up will give you different characteristics of the intermediate filament. However, the really big difference here is that our monomers are filamentous, they are not globular. So again, if we go back and look at, I don't even know where I was at anymore. This was, again, our microfilament. We have these globular monomers that then get polymerized out to make this nice long individual strand. And so this is not what's happening with the intermediate filaments. The monomers are filamentous, not globular. So we take out one filamentous monomer, and we essentially wind it around or weave it with or coil it with another one, and we get this thing called the coiled dimer. We take two coiled dimers that will then end to end. So let me draw things because I'm no good at that. Okay, so we have a filament. We get two coiled together. We get a filamentous coiled dimer, and then we'll put two of these together. I'll have an illustration of this. That's horrible. Get two of these bound back to back like this. We get tetramer.
They bind at the end.
What's that?
At the end.
Yeah, at the ends. On the narrow side. And then we get another one kind of like this, another one kind of like this, and then we get essentially two of these on top of each other, and then we get a protofilament. We take 8 protofilaments, form them into a cylinder, and we get the intermediate filament. So hold on a second, I'll show you what we're talking about here in just a minute.
What was the correlation between the tetramer and the
What's that?
What was the correlation between the tetramer and the
So the protofilament, okay, so imagine I have so here's my tetramer, okay? I'm now just going to represent that with a line because I can't draw. And then we have essentially these polymerizing like this. Overlapping, but essentially you have 2 tetramers thick. Wait, hold on, I'm doing this wrong. These are not end-to-end. They're kind of like that. So they're almost end-to-end, but they're actually overlapping each other somewhat. So then you have something that looks kind of like this. And then you have multiple of these end-to-end. So the tetramers are then end-to-end.
So the protofilaments overlap the same ways, in the same way as you did to get the tetramer, but they're overlapping tetramers?
Yes. That makes sense? Okay. If it doesn't, let me know. So this is a tetramer.
And that equals 1 line, not 2.
What's that?
That equals 1 line.
No, this is 2 lines. So equals that. Is there different colors here? So then, to get the protofilament, we go to this, where this is one tetramer here, this is another tetramer there, another tetramer there. So the entire tetramer is end-to-end. Let me show you this way, that's drawn better. So we have a filamentous monomer. It's coiled with another monomer to make the dimer. It is overlapped with another dimer to make the tetramer. And then end to end, of the tetramers, we get a protofilament. And then 8 of the protofilaments in a tubular structure is the intermediate filament. Way. So we have, for microtubules, we have other proteins associated with them called MAPS, microtubule associated proteins. In microfilaments, we have proteins that bind to that. They're MBPs. The microfilament binding proteins. Can anyone take a wild guess what the proteins associated with this would be called? Very close. Intermediate filament associated proteins. I did not name these. Do not blame me. These are called intermediate filament associated proteins, or IFAPs. Moving on.
Wait, I didn't get it yet.
You didn't get IFAP, okay. I'll wait for just a little bit longer. I really need some tea right about now. I'm sure there'll be something about the test, on the test, about intermediate filament associated proteins. Okay, so there's 5 classes of these different ones. They're different based on their structure and then also their function. However, not all of their functions are completely, totally clear. They tend to be one of the harder things in the cell to study. I'm not exactly sure why, actually. I've just read that several places. I've never tried to study them myself before, so I don't really know.
5 classes of IFAPs.
Yes, there are 5 classes of IFAPs. IFAPs. Let's go through a couple of them that we do know. Vimentin. Vimentin is in all cells. Yes. Go back. Was that a little bit too fast for that? Okay. What's that?
Is there a reason Vimentin started with C?
What's that?
Started with like point C.
Oh, yeah, because I don't know. These at one point these were all kind of like outlined, and then like the outline kind of getting, and then some of them have retained like the outline numberings, and some of them have not. So some of them still have like vestigial outline numberings. So they're not C vimentins, they're just, this is point C vimentins. Does that make sense? However, that being said, this is also C.
Yeah.
So I don't
Not
know.
organized. But that's okay.
Yeah. So judgy.
No, it's okay. It's good.
C, vimentin. So vimentin is one of these. This is found in all cells, and the primary function appears to be to provide a scaffold and structure for the cell itself. And so a lot of what we're actually making with the cell structure is this. Also, keeping some sort of intracellular organization as well within the cell. So, for instance, we will place organelles on these and place organelles appropriately within the cell. Excuse me. For instance, Muscle cells will often have a load of mitochondria in one side of the cell. That's particularly important because this is where your cell muscle contractions start. And so we will have a lot of mitochondria there down in one end. And vimentin is one of the things that provides the scaffold where we can keep mitochondria in one place in the cell that we want it to and not have it just kind of floating around willy-nilly within the cell like, some kind of vagrant. Also, they appear to work somehow in cholesterol transport throughout the cell as well, although that seems to be less well understood. Another one that we have is called desmin. This is particularly in muscle cells, and they are important in what's called sarcomere organization. Sarcomere is actually the arrangement of cytoskeletal elements that are repeating inside the muscle cell that actually is kind of just the organization of the contractile apparatus. Principally in the contractile apparatus is myosin and actin microfilaments in there. And so desmin organizes these actin-myosin complexes. There is a disease called desmin-related myopathy. I was going to talk a little bit about that, but no, I'm going to skip over that for the moment for time reasons, because if you looked at the syllabus lately, I'm about a week behind. I'm sorry. We also have GFAPs. These are glial fibrillary acidic proteins. These are another kind of intermediate filament. And these provide strength and structure to the cell as well. We first found them in glial cells. Glial cells are, in your brain, they are not neurons, but they are essentially support cells that help neurons actually carry out their functions and do a lot of other things as well. There's other classes that you can learn lots about glial cells in. And so really we're just providing, yes?
You said these are the intermediate,
This is a class of intermediate filament.
they're not regulatory,
What's that?
not regulatory proteins.
No, these are not, well,
What about,
so there are intermediate filaments along with their IFAPs, yes. What's that?
these are comprised of like intermediate.
Intermediate filaments plus, yes.
IFAPs.
Yeah. And so by different combinations of different types of monomers of the I was saying microfilaments, I meant intermediate filaments, plus their associated proteins, you get different kinds of intermediate filaments and different structures and functions of those. And then also associated with neuronal tissue, we have neurofilaments. These are particularly in axons. So axons, if you look at a neuron, my bad drawing, get out of here. I need more bad drawing. If you look at a really basic structure of a neuron, it has a cell body where your nucleus is. You have this long, kind of skinny structure called the axon, and we have dendrites out here. And essentially how a neuron works, here too. Information comes into the dendrites or actually into the cell body. Those signals get kind of synthesized and processed in the cell body, and then they're sent on to the next neuron or to whatever other cell that needs to go to by this axon. So you get this electrical pulse called an action potential that's going to travel down this axon. Now, some axons in you are very, very incredibly long. You actually have some axons in some nerves that are like a meter long in you. That's a really long cell. They have to travel somewhere from your head, and some of them travel all the way down to like your upper leg area. The ones that travel down to the bottom of your feet actually come from the base of your spine. So you have some that are really long inside you. And they're really skinny. And so it'd be really easy for those cells to break. In neurons, I mean, hey, if you started having neurons breaking all the time, that'd be bad. And so we have these specialized neurofilaments that are in axons that provide strength and rigidity and helps maintain the 3D structure of this axon. So these are really important for maintaining the axon integrity, and maintaining axon integrity really helps you with transmitting signals throughout your body. It actually turns out that breakdown of these neurofilaments play a role in ALS, which is amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. To make a long story short, we have problems with these that cause the neurofilaments to break down. They're not getting transported all the way down the axon like they're supposed to, and not providing the structure that they are. So you get buildups of neurofilaments in some places in the axon, or in it's called the axon hillock here. You might have like this bunch of neurofilaments here that all kind of get backed up. Not enough down here, so you start getting collapses in these axons. Not really with that sound. And then you start being unable to transmit signals down these axons anymore. This is particularly bad because, this principally happens not in all neurons, but principally starts happening in your motor neurons, and you lose control of your ability to move around and stuff. That's bad. We have these other ones, also known as cytokeratins. These are, yes, Ryan.
Does the breakdown of the neurofilaments actually help the cell? Does it just render it incapable of functioning correctly, or does it actually break it down like the entire cell?
So normally the neurons are still there, but yeah, it prevents them from functioning correctly. You can't actually transmit an action potential down the axon anymore. So, it might as well be dead. But I think they stay alive, yeah. Okay, so cytokeratins are intermediate filaments that that are very common in epithelial cells, particularly also with structures associated with your epithelium, such as skin, nail, hair, claws, fur, those sorts of things. These are really, really strong. So one thing that you probably have noticed is kind of being a recurring theme is that a lot of different intermediate filaments are important to maintain mechanical strength of the cells, and that's no difference here. These are really actually some of the most strong binding of best intermediate filaments for providing mechanical strength. If you think of like most of your body compared to your hair, even though it's you can break it off fairly easy for its size, is pretty strong stuff compared to like a lot of other things. If you were all made out of hair, you would be really tough. It's kind of a weird thing to say. I don't know.
Hairy people are tougher.
Probably. I think hairy people are like way tough. I see a really hairy person, and I get scared. Which then is like most mornings looking in the mirror like, whoa, that's scary. But for other reasons other than that, it's not just that scary. It's lots of reasons scary. I frighten myself sometimes, oftentimes. Anyways, okay, beside the point. Anyway, so one of the particular things that make these particularly strong is that we have numerous disulfide bonds, both in between the monomer filaments, the monomer filaments, but then also between the protofilaments as well. And so these common disulfide bonds actually create a good amount of strength, and so yeah, that's good. One thing that's just kind of, as I talked about this a little bit before, but I'm just going to talk about the first one, and we're going to pass through the rest of it. But since you have all these disulfide bonds between these intermediate filaments in your hair, one of the things that when you get perm, You need to break those disulfide bonds and reform those intermediate filaments. And so you put in the hair bathing recap with ethanol, which breaks those disulfide bonds, and then you shape the hair the way you want it. Get that out, disulfide bonds reform, and so that you get hair in a different shape than you had before. I'm not going to worry about a lot of these because, again, I do want to, at least by Friday, get a little bit to cell junctions, which actually is right now. So, it gives you a couple minutes. I will leave it at that for now then. We will talk about cell junctions on Friday. Enjoy your time before your final class in here, and then also before your finals.